suffix trichus, so this is monotrichus, right? What if, you guys, we have flagella at both ends? Amphitrichus, right? What if we have a tuft, a bundle of flagellum at one end? That's lophotrichus. And my favorite one is, you remember this one, you guys?
No.
That's peritrichus. Peritrichus, okay, awesome, you guys. You guys all right with that? Okay. Peri, P-E-R-I, like all around, you're just covered in, good. And folks, what about E. coli serotype 0157H7? What is the H antigen? And remember, we're chapter 9 motility. It's the flagellar antigen? Good, the flagellar antigen, awesome, you guys. Okay. Oh, I know, you guys. Make sure. Do you remember how we did motility auger? So you went straight down and straight back up, you guys. So if a microbe is motile, these aren't the best, but oh well. Oh, there's a better one, you guys. So if this is microbe A and this is microbe B, which is motile? A, B, both, or neither? B, right? Because remember, you guys, if the microbes are motile, they'll grow along the stab line, and then they start using up the nutrients. We have toxic metabolic waste products, so they're going to move away from the stab line into the auger if they're motile. If they're not auger, they're just stuck there, right? Okay, so remember motility auger. Oops, good. Okay. All right, you guys. So that was chapter 9. Chapter 10 was a big one. That was microbial growth media. Okay, so you guys do remember, big one, being able to classify the medium. Is it all purpose? Is it enrichment? Is it selective differential? And so you guys also, we started out with the airborne microbe experiment. So as we said, you guys just remember that if, say, at 30 minutes we had, I'll make this up, 100 colonies, we would predict it 60 minutes we would have twice as many, like 200 colonies, just in general. Yeah, and you guys, there will be a question on one of the airborne plates. I'll circle a colony and have you describe it, like measure it, describe the shape, describe the pigment, right? And then also, you guys, on the airborne microbe, we use TSA. Can you classify TSA? All purpose. You're gonna throw a wide range of bacteria and fungi. And what about Saccharomyces dextrose? How would you classify that? Selective for fungi. So most of the colonies growing on our sap plates, you guys, they would have been either yeasts or molds. And do you remember how we inhibit bacteria in Saccharomyces dextrose medium? Low pH, right? And really high glucose. Good job, you guys. Okay. Then you guys, we move on to, okay, then you guys will move on to, how about blood auger? You guys, how would you classify blood auger? Is it all-purpose, enrichment, selective differential? It's enrichment to grow fastidious bacterial pathogens, right? And what else?
It's
Differential based on homolysis, right? So that'll be important, guys. So we talked about 3 types of homolysis. What is total clearing, you guys?
Beta.
Like the contents of the red blood cells are totally destroyed. Beta, right? Can you name 3 beta hemolytic gram-positive coccal pathogens that we mentioned? So Staphylococcus aureus, right? So think of abscess. Streptococcus pyogenes, you guys like strep throat, but they can also cause abscesses, right? And what if you have a pregnant mom? What do you want to culture? Vaginal mucosa, looking for group B strep, right? Because that can cause a neonatal sepsis and meningitis. Okay, so you guys, let's say I give you a station, and let's say maybe it's an abscess, you've isolated gram-positive cocci, beta hemolytic, and you're trying to decide between Staphylococcus aureus and Streptococcus pyogenes. What simple benchtop test could you do to distinguish between Staphylococcus and Streptococcus? Catalase, right. Good, you guys. And who's gonna be catalase positive? Staphylococcus aureus, the whole gene is Staphylococcus. And if it was a Streptococcus pyogenes, you guys, or a group B strep, what would the catalase test be? Negative, awesome. Excellent, you guys. Good. Excellent, you guys, excellent. Okay. What if you have a pathogen and plate it onto blood and it causes a dark greenish discoloration? What kind of a hemolysis is that? That's alpha hemolysis. Okay. Can you name 2 gram-positive bacterial pathogens, gram-positive cocci, that are alpha hemolytic that we mentioned?
Oh, Streptococcus mutans?
Good, Streptococcus mutans, right? And what about
Streptococcus
pneumonia? Pneumonia, good. And those 2 Streptococcus, you guys, if you mixed them with hydrogen peroxide, what would you see? Nothing, right? Because remember, members of genus Streptococcus, no catalase, you would see no bubbles. Good job, you guys, excellent. What if there's no hemolysis? What do we call that? Gamma, good. Okay, so you guys, let's say that you've got a beta hemolytic gram-positive coccus. It's catalase positive. And now you're trying to figure out, is it Staphylococcus aureus or its cousin Staphylococcus epidermidis? What selective differential media could you use that we discussed? How about MSA? What does MSA stand for? Mannitol salt agar. Okay, it's selective how, folks? What is the inhibitor in MSA? What does MSA stand for?
Mannitol salt.
Agar, right? So what's the inhibitor?
Salt.
Yeah, and exactly how much sodium chloride? 7.5%, right, weight per volume. That means 7.5 grams per 100 mils, right? So you guys, if I want you to make a liter of MSA, how many grams of sodium chloride are you gonna add? If it's 7.5 grams per
750.
100 mils,
Oh,
you're gonna multiply
75.
by
70.
10, right?
75.
To get, how many grams? 75 grams, right, of sodium chloride. So who will that inhibit? Microbes that aren't salt tolerant? I know, that sounds so silly, right, you guys? But remember, members of the genus Staphylococcus are usually, they've evolved to live in high salt environments like the surface of our skin, right? So we'll select for like salt tolerant members of the genus Staphylococcus, yeah? But you guys, mannitol salt agar is also differential. What is the ingredient, or what are the 2 ingredients that make MSA differential? That's right, the M, the mannitol, the sugar alcohol, just pretend it's like a sugar, right? So we can look for mannitol fermentation. And what's the pH indicator? Default pH indicator, you guys? Phenol red, right? Phenol red, good. So you guys, if the microbe can ferment mannitol, acids will be produced. So what color will the phenol
Yellow.
red turn?
Yeah.
Yellow, right? And if the microbe can't ferment mannitol, it will tear apart amino acids, making an alkaline reaction. What color will phenol red turn?
Red.
That deep red, right? So you guys, here's the question. Can Staph aureus ferment mannitol? Yes. So you guys, on this plate, which is the Staph aureus side? The yellow side, right? Staph epi can't ferment mannitol. So Staph epi is growing over here giving this alkaline reaction because it's tearing apart the amino acids. Okay, does that ring a bell, you guys? Good, awesome. Okay, fecal contamination of food. Oh, sorry, Sean. Yeah.
Staph aureus can ferment mannitol?
Yes, can. Yeah. So our Staph aureus on mannitol salt agar is going to be nice and yellow. Yeah.
What is the anatomy?
I'm sorry? Say again?
What is the another?
Staphylococcus epidermidis. Yeah, the cousin, less aggressive, can't ferment mannitol, so it'll have a nice alkaline red reaction on MSA. Okay. So you guys, you know, I'm obsessed with fecal contamination, right, of food and water. So you guys, what if we send you up to Butte County and you're trying to find out what's the source of the norovirus that's making everybody sick? So you know it's fecal, probably most likely fecal contamination, right? So you're sent up there to try to figure out where's the fecal contamination coming from. So you're taking food samples and water samples. You've got all these samples. So what kind of selective differential media could you choose that we've talked about that would help you quickly screen for the presence of our fecal indicator? Could you use? Could you use McConkey's? Does that ring a bell, you guys? So McConkey's is gonna inhibit non-enteric bacteria. It's gonna select for enteric bacteria like our fecal indicator E. coli, so that's really good, right? And furthermore, you guys, McConkey's is awesome. It's differential. It lets us differentiate between microbes that can ferment lactose and those that cannot. So remember, you guys, fermenting lactose is kind of an unusual property. Does E. coli, our indicator of fecal contamination, ferment lactose? That's important to remember, you guys. E. coli ferments lactose, right? Because remember, it's part of our mammalian intestinal bacteria, right? And as mammals, right, we make milk for our babies. So remember, E. coli does ferment lactose. So you guys, in McConkey's, the single sugar is lactose. And the pH indicator, this is a tough one, you guys. I always forget it. The pH indicator is neutral red. So neutral red at low pH, acidic pH, is going to be pink or red. So you're going to have pink or red colonies if you have lactose fermenters. And it will be colorless at pHs other than acidic pH. So you guys, if you have a water sample, you plate it on McConkey's and you incubate it and you pull it out the next day and there's all these pink colonies, would you be potentially alarmed? Yes, because who could the pink colonies be? Could be E. coli, right? And so that suggests if it is E. coli, you know, we found one of the sources of fecal contamination. Now remember, you guys, how we said, but wait a minute, E. coli has this cousin, Enterobacter. And Enterobacter behaves a lot like E. coli. Enterobacter could be just from dead plant material. Which tests could you run to verify the colonies growing are E. coli?
Cat out, right?
How about the InVid test, right? So you guys, if the colonies growing on McConkey's are E. coli, you run the indol test, will it be positive or negative? If it's E. coli, it should be positive. What color? Red. If you run the methyl red test, if it's E. coli, what color should it be? Red. It should be red, which is positive. Good. If it's E. coli, you run the BP test. What color should it be? Not red. It should be negative for E. coli. If you run the Simmons citrate auger, you guys, if it's E. coli, what color will your Simmons citrate auger slant be? Not blue, right? Green. So remember, if it's E. coli, InVid positive, positive, negative, negative. Right? Okay, good job, you guys. All right. Okay. That's, I mean, and again, you guys, I'm not, obviously I can't hit everything, but I think for chapter 10, we at least hit a lot of it. Okay. So now you guys, chapter 11 was aseptic technique, chapter 13 was isolation. So probably what I do, you guys, is maybe for aseptic technique, have some plates or broths or slants, and maybe ask you, do these appear sterile or do they appear contaminated? Something really simple like that, right? And for isolation, guys, a street plate, I'd ask you to see, do you think this is a pure culture or not pure? And remember, if it's not pure, you're going to see different types of colonies growing, right? Also, you guys, I could ask, because I did see this on some of the clinical sample re-street plates. I could have a street plate. And remember, you guys, on the street plates, our goal is to get isolated colonies. And what if I had a street plate, you guys, at the station, and say it's been streaked and incubated, and there's no isolated colonies? can you come up with 2 hypotheses as to why, say it's your partners, right? Why there's no isolated colonies? What are 2 really common mistakes that we make when we're streaking plates for isolated colonies? Okay, and I mean, so that was such a good thought, you guys. If your loop is too hot, you're gonna have no growth, right? Right, so think about, no, I have too much growth. What are 2 really common mistakes? Could I, and this is so common, you guys, we think a little bit's good, more's gotta be better. Could I be going double dipping for every quadrant? I'm going back in and getting more of my micro. That's a real common mistake. And a second really common mistake, you guys, is we forget to do what? Yeah, we forget to flame between quadrants. Okay, those are real common mistakes. Okay, good. Alrighty, so that's 11, 13. 14, control of growth, you guys, so, oh yeah, sorry.
Professor, can you quickly go through what heteropod do you need?
Yeah, and I can do that, you guys. Would you prefer that we do that, do the arthropods and the helmets? I'll do whatever you guys want me to do. I'm here for you to command.
Just the heteropods.
Okay, so help remind me, you guys, that after we do arthropods, we are at control of growth, but if we run out of time, you guys remember that practice chapter 14 quiz in the study guide packet, that will help you a lot. Okay? And just, let me just do a couple really quick, because I'm afraid, okay, so you guys, do you remember which antiseptic or disinfectant was best at inhibiting both our gram-negatives and our gram-positive bacteria?
Tincture of iodine.
Tincture of iodine, good, you guys. And the iodine is what? A halogen?
Oh,
okay,
yeah.
so again, you guys, try to do your practice quiz. And then the other one, you guys, I just want to emphasize is
alcohol.
if you're dealing with a blood spill where there might be HIV, hepatitis B, or hepatitis C, and you want to disinfect the inanimate object after you've cleaned up any organic matter, feces, vomitus, tissue, blood, what does the CDC recommend to inactivate HIV and Hep C? 1 to 10 freshly diluted bleach. Good job, you guys, excellent. Okay, and now, okay, so we'll switch to chapter 21, folks. Okay. So remember, chapter 21 was helmets and arthropod vectors. So folks, yesterday when we were going through this, we were trying to remember how we're always saying, if you can't, like you're tired, you can't remember, it's always going to have a default answer. It's always going to have a default answer, or maybe when we're trying to classify organisms, if we can just remember, well, most organisms are fit, so all I need to do is memorize the exceptions. That might be with classifying the helmets. Because you guys remember, helmets, we said there's 3 groups. The nematodes, the roundworms, the cestodes, the tapeworms, and the trematodes, the flukes. So you guys, all of the helmets we looked at, they're all nematodes except for one. Can you remember the exception? Taniosolium is the exception. Taniosolium is a cestode. Everybody else was a nematode. Okay, all right. So you guys, do you remember who this guy is? This is Ascaris, nematode, cestode, or trematode. Remember, default, you guys, if you don't know, just say nematode, and what? 80% of the time you'll be right. So Ascaris is a nematode. What's transmission? Fecal oral, right? So fecal oral, the eggs are going to be in the feces when you swallow them. Usually, at low numbers, it's not that big a deal, but what could it cause, you guys? What's a problem with Ascaris sometimes? There's so many, they block the intestine, right? Yeah, and remember the photo, you guys, of the little child that was undergoing surgery and the adults were coming out the mouth and nose? Yeah, okay. And then, folks, okay, ooh, this is a good one. So Ascaris is fecal oral transmission. Is there another helmet that we studied where there's also fecal oral transmission? Taniosolium, good. What's the common name? Or tapeworm. Okay. Why, you guys, is fecal oral transmission of Taniosolium eggs so important? What horrible thing could develop? The neurosis. Remember those those sections you guys through the brain where all the little cysticerci were in there. A leading cause of epilepsy, right? That's just wild. What's the second way we can get infected with Taeniusolium?
Oocysts.
Undercooked pork, right? Where we're actually eating the cysticerci and then they become adults in the intestine. Good job, you guys. Taeniusolium, is it a nematode, a cestode, or a trematode? Cestode, good, you guys, excellent. Okay, what was a helminth that was transmitted by mosquitoes? Wuchereria, good. Wuchereria bancrofti. And you guys, why is chronic infections with Wuchereria so devastating? What does it cause? The elephantiasis, the lymphatic filariasis, remember the swelling, you guys, of the limbs, the scrotum? Good. And you guys, how would you diagnose filariasis in your patient? What kind of sample are you gonna take? What are you gonna look for? Blood sample, right? And think, you guys, mosquito-borne, right? The mosquitoes are getting it in the blood, right? And we're gonna look at it, and which stage are we gonna look at in the blood smear? The microfilaria, awesome, excellent, you guys, great. So you guys, we said that Taeniusolium can be transmitted through undercooked pork. Is there another helminth we studied that can also be transmitted through undercooked pork, undercooked bear, undercooked walrus? Trichinella spiralis, right? And remember, you guys, the diagnostic sample we show you is muscle, stained muscle, with the little larvae, like little cinnamon buns in there, right? Food analogies are always good at remembering these things, right? And they can be really devastating. They can cause damage to muscles. They can mimic heart attacks. Good. Okay, let's see here. And I forgot. Oh, and you guys, we said the pinworm would be a bonus question. Enterobius. It's just the most common helminth infection. I think probably worldwide, and usually they don't cause that big a problem. It's just, okay. Let me see here. Is that all of our homes? I think so. Good. Okay, good, you guys. And then, folks, with regard to vectors, what it'll be matching. It'll be matching. So I'll have like photographs of the vectors. They'll be letter labeled. And then I'll have you match which pathogen they can transfer. And again, you guys, it's just limited to what we've talked about in our lab. So you guys, and I know this is really hard. Okay, so you guys, here's a photograph of an arachnid. It has 8 legs. It's a blood feeder. The common name is a tick. So which bacteriospirochete do ticks transmit here in Northern California? Borrelia burgdorferi, awesome, you guys. What's the name of the infectious disease? Lyme disease, good, you guys, excellent. And do you know the scientific name of the tick that transmits Borrelia here in Northern California? Good, Ixodes pacificus. Good job, you guys, excellent. And then, okay, so that's the arachnid. The rest are insects, you guys. So here's a little guy. That's the common name is a mosquito. Can you name a virus that mosquitoes transmit here in Northern California? Well, it's not a virus. Are there other viruses that mosquitoes transmit?
Yes.
Like Zika, right? And Zika we're worried about, you guys, because if mom's pregnant, then there's transplacental transfer, and the Zika invades the neurons and decreases neuronal development. Good. Any other viruses, you guys, we should be dengue, right? Chikungunya. Good job. What about a protozoal pathogen that mosquitoes can transmit? Plasmodium, good. Malaria, good. What about a helminth that mosquitoes can transmit? Wuchereria, good job, you guys, excellent. Okay, so you guys, here's a micrograph. Our cats and dogs have these. They're really tiny. They go boing, boing, boing. Those are fleas, right. Can you name a bacterial pathogen that fleas transmit here in California? Yersinia pestis, which causes bubonic plague. Good job, you guys. This is hard to see, you guys, if we were maybe visiting in Africa. These are very aggressive blood-feeding flies that will chase us to feed on us.
Sure.
Who's this guy? So the name of the fly is Glossina, or the tsetse fly. And which protozoal pathogen does Glossina transmit? Trypanosoma brucei, which causes African trypanosomiasis or sleeping sickness. Good, you guys. And here's one we're worried about here in North America. The common name for this one is the
Kissing
kissing
bug.
bug. This is Triatoma or reduvids. Which protozoal pathogen does Triatoma or reduvids transfer?
Trypanosoma brucei.
brucei, which causes
Chagas disease.
Chagas disease or American trypanosomiasis. Good job, you guys. All right. Does that feel semi like we did that? Okay. We'll put pictures. So it will be important, you guys, that you can identify what the vector is, because it will be photos. It'll be photos of them, and they'll be letter labeled, and then you'll match them to the
Will
pathogen.
it just be the photos of what you say?
No, there will be no description. It'll just be a photo. Yeah, just kind of like in chapter 21, where it has you looking at slides of the different vectors or samples. That'll kind of be the same thing. Yeah. Okay. Yeah. So you guys, we're at time right now, and I know you're probably just feeling exhausted and overwhelmed. What I would really recommend, again, you guys, chapter 14, use that practice quiz in the study guide packet, okay? And I know you guys, it's not all that helpful, but like the chapter 16 and 17, we covered all the topics today, and I know it's just a lot. And I hope you guys had a chance maybe to look at some of this stuff over the break. I hope. So, be of good cheer, you guys. Have faith in yourselves, and just do the best job you can.